The genital or reproductive system. We have to talk about both the male and female genital system. First, we'll be starting with the male description, where we'll be focusing upon the details of the seminiferous tubules in the testes, the location where we have the formation of sperms, the spermatogenesis. We also need to talk about the blood-testis barrier and some details about the epididymis, taking into account some of the cells and the hormones as well. Testis is the male gonad, and we understand that it is having a connective tissue covering which is called as the tunica albuginea, and inside the testis, we are having seminiferous tubules lying between the various septations which are sent by tunica albuginea, and those seminiferous tubules, they will be involved in formation of the sperm which is called as the spermatogenesis, and along with these seminiferous tubule, we have got some cells in the testis whose details we need to discuss. Like you can mention, there is one called as the Leydig cell, the interstitial cell of Leydig lying in the interstitium in relation to the seminiferous tubule, and then there is another one which is in the seminiferous tubule called as the Sertoli cell. The Sertoli cell is also called as the nurse cell because it is the one which is going to support and provide nutrition to the developing gametes. So Sertoli cell is supporting the cells which are involved in gametogenesis, the spermatogenesis, like the gametogonia, the gametocyte, and it is giving nutrition to them. Plus, it is also providing the protection, especially when we are talking about the blood-testis barrier. And how is it helping to form the blood-testis barrier? We need to discuss. At the moment, we can mention the Sertoli cell give us two hormones as well, known as the Müllerian inhibiting substance and the inhibin. This Müllerian inhibiting substance is going to inhibit the Müllerian duct during development so that female reproductive system is suppressed and the male reproductive sexual characters will be evolving. Whereas the inhibin is actually working like an inhibitory hormone upon the pituitary. You know, all these hormones are basically under the control of pituitary gland. It is the pituitary gland which is giving us some of the hormones which work upon the gonads, like it is giving us luteinizing hormone, follicle stimulating hormone, and we should know it is the luteinizing hormone which will be controlling the Leydig cell. So pituitary gland, the master endocrine gland, is giving the luteinizing hormone which is affecting the Leydig cell. Leydig cell themselves will be giving the testosterone. This testosterone is important for the reproductive function. For example, the secondary sexual characters are determined by the testosterone. Then we have mentioned about the Sertoli cell giving us the inhibin. Inhibin is actually a feedback mechanism hormone upon the pituitary gland, and using this feedback mechanism we can control the secretion of luteinizing hormone and follicle stimulating hormone from the pituitary. So it is basically a loop to maintain the balance between the concentration of luteinizing hormone, follicle stimulating hormone, and the testosterone in effect. Now let us look at the diagram of testis and discuss some details of the seminiferous tubule. Here we can see we are looking at the testis. Let us zoom on this area first before we look at the histological features. We mentioned that the testis is the male gonad covered by the tunica albuginea, which is sending the septations towards the interior. So these are the septa separating the seminiferous tubules, and the connective tissue is named as the tunica albuginea, and within the septations, as we are magnifying, this will be the area of the seminiferous tubules. Now before we look at the seminiferous tubules, we have to understand that the sperm, as they are formed in the seminiferous tubules, they are non-motile. They are being pushed further with the help of some myoid cells, and these myoid cells will be pushing the sperms towards what is called as the epididymis. In this epididymis, they will be gaining the motility. The sperms gain their motility in the the epidermis, where they are stored also and matured also. So maturity, motility and storage happens in the epidermis. Later they'll be pushed into the ductus deferens. Now since this is a topic where we have to focus upon the histological features, let us magnify this diagram so as to look at the details of seminiferous tubules and the cells related with it. So as we magnify the seminiferous tubule in the testis, we can find in this seminiferous tubule we are having the process of spermatogenesis going on. This is one of the seminiferous tubule which have been magnified here, and in the vicinity you will have few others as well. The structures which are in the surrounding, you can see there are some blood vessels which you can mark here, and along with the blood vessels we can see there are some interstitial cells as well. Interstitial cells of the Leydig. As we have mentioned, they will be giving us the hormone testosterone under the influence of pituitary gland, the luteinizing hormone. So pituitary gland will give us the luteinizing hormone, and under that the Leydig cell will give us the testosterone. We'll be talking about these cells in detail, but at the moment let us focus upon the cells which are related to the seminiferous tubule. In the seminiferous tubule we find that towards the periphery we have the germ cell, primordial germ cells, and we know the primordial germ cells, they are remaining dormant until puberty, since we understand the process of spermatogenesis begins only after puberty. So it is also understood that if the sperm formation begins after puberty, then the sperm which is being formed here, say this is primordial germ cell which is now making the spermatogonium, making the primary secondary spermatocyte, and these sperms are formed quite late in the lifetime, and they are just like a foreign body to the immunocompetency of human body. See, the immunocompetency has already developed. The thymus, the bone marrow, B, T lymphocytes, they are immunocompetent already. It's puberty, and at puberty you are having a new type of cell formed. So the component of the blood vessels, they will be immediately identifying the sperm as a foreign body, a new antigen. They can mount a response and kill the antigen. There can be autoantibodies which will be destroying the sperm. That is why we should have a blood testis barrier. You cannot expose the sperm with the component of the blood, and for that blood testis barrier we have some cells. We were calling them as the sustentacular cells. So these sustentacular cells or the Sertoli cells, what exactly are they doing? How do they form the blood testis barrier? We will look into a diagram, but these sustentacular cells, they are having some lateral processes, apical processes. They are quite large in size, and one sustentacular cell is connected with the other sustentacular cell, making tight junctions, and because of those tight junctions, the blood vessels, the component of the blood cells, cannot be exposed towards the lumen of seminiferous tubule. Plus these sustentacular cells, as we have mentioned, they have several functions. Not only they provide protection by this blood testis barrier, they are also providing nutrition and support to the cells involved in spermatogenesis, maybe the germ cell or the spermatogonia, spermatocyte, or the spermatids themselves. Plus they give us hormone also. We have discussed the Sertoli cell will give us two hormones. One of them is the Müllerian inhibiting substance and the other was inhibin. This inhibin was having a feedback mechanism upon the pituitary, so as to see there should not be excess secretion from the pituitary gland in terms of luteinizing hormone or follicle stimulating hormone. Now what about these Leydig cells? How do you identify the Leydig cells? First of all, they are outside the seminiferous tubule. They are in the interstitium. That is why they are called interstitial cells of Leydig. Plus they are very large. You can see very large cells and having pinkish color because they have high amount of smooth endoplasmic reticulum. So eosinophilic, take more of eosin, and they have lipid droplets also. Polygonal cells with a lot of lipid droplets, large cell, pink cell, and secreting hormones like the testosterone hormone. Now let us magnify the sustentacular cells making the tight junction among each other so as to create the blood-testis barrier and preventing the exposure of the sperm from the blood vessels which are lying outside in the interstitium like we have seen here. In this diagram, we'll be talking about the blood-testis barrier, and as we are magnifying the diagram of testis looking inside one of the seminiferous tubule and still magnifying it, we find that the sperms are being formed towards the interior, towards the lumen of the seminiferous tubule, and towards the exterior we are having some interstitium. There will have the blood vessels, and we want to make sure that whatever the contents are there in the blood vessels should not get exposure to the spermatids lying on the interior. And for that purpose, we are having these big cells, one shown here, one shown here, the Sertoli cells. As we have mentioned, the Sertoli cells, they have three functions. Number one, they will form the blood-testis barrier so as to provide the protection. Number two, they are supporting the cells involved in gametogenesis, especially spermatogenesis here. And number three, they provide the nutrition. In addition to the various hormones they were providing us. Now let us demarcate one of the Sertoli cell here, which is quite a large cell coming from the base towards the luminal side, and another Sertoli cell there. You will see as we demarcate them, starting from the base, they are having some lateral processes. You can show it on both sides. One here, then another one here, third one here, and these large cells are reaching the luminal side as well. Now this is one of the Sertoli cell. Let us demarcate it on the other side as well in different color. So this is another Sertoli cell giving a lateral process, another lateral process. Then they will have the apical processes also. In the apical processes, as you can see, they are holding the spermatids. And then we can mention two compartments here. One of them is called as the basal compartment and the other is adluminal compartment. So towards the base, we have the basal compartment, and towards the lumen, we have adluminal compartment. At the apical processes, they are holding the sperm, as you can see, towards the luminal side. But then these lateral processes, like you can see, they are holding hands with the next Sertoli cell and making tight junctions. As you can see, there are some junctional complexes which will not be allowing certain components lying outside towards the interior. So these are the junctional complexes, as you can see, making the blood-testis barrier. Now these junctional complexes, they are semi-permeable. They will be allowing the cells involved in spermatogenesis to move inside as they keep developing. Say for example, here you have got the spermatogonium. It can move through the junctional complex interior to become primary spermatocyte, becoming the secondary spermatocyte, and then becoming the spermatid. So these cells are allowed to move towards the interior through these cell junctions. They are semi-permeable to them, but these junctions will not allow any component from the blood, like any lymphocyte or antibodies, to move inside, hence creating a blood-testis barrier. Let us draw this diagram ourselves now, and there it will become more clear. As we draw the histological diagram of the testis, we can mention these are the seminiferous tubule, one on either side, and lying between the seminiferous tubule, we are having the interstitial cells of the Leydig. Now, these interstitial cells of Leydig, they have large size, are polygonal, and will be appearing quite pinkish because of the presence of the large amount of smooth endoplasmic reticulum. And they will also have the lipid droplets. As we know, they are going to give us the hormones, the testosterone hormone. And then we can move on to the details of the blood-testis barrier. Let us draw two of these Sertoli cells inside the seminiferous tubule, having the lateral processes and the apical processes as well. They are quite large cells, columnar in nature, and we are drawing one here, one on the other side. So one Sertoli cell here, and we draw one Sertoli cell here, and we see their lateral processes. They are holding on to each other, making what is called as the tight junctions. So here we are having the tight junctions, and they are semi-permeable. As we will see, the cells which are involved in spermatogenesis, they are allowed to move from the basal compartment towards the luminal compartment. Say for example, we are having the primordial germ cells toward the basal surface, and these primordial germ cells, they were allowed to move inside. Now they have become spermatogonium, which then become primary secondary spermatocyte, and finally finally become the spermatid, attaching to the apical processes. So we have got primordial germ cell becoming the spermatogonium, becoming the primary, and the secondary spermatocyte becoming the spermatid. In that process, as they keep on jumping to the interior, that is possible. These tight junctions allow them to move inside. Semi-permeability is there. But then these tight junctions, they do not allow any component of the blood vessel. Like you see, there are some cells in the blood vessels now, or maybe some toxins as per se. Those toxins cannot move towards the sperm because of these tight junctions. As it is understood, the process of spermatogenesis begins after puberty, and these sperms are basically something new for the blood cells. They can mount an immune response, form autoantibodies, destroy these sperms. So we don't want that to happen. Like it may be happening when you are doing the vasectomy. Some of the sperm cells may come out exposed to the blood. Then there'll be autoantibodies and destroying all these sperms. So normally it is being prevented by these Sertoli cells. These Sertoli cells, not only they are providing the protection, they also provide the support and they provide nutrition for the cells involved in the process of spermatogenesis. Now what you get here is called as spermatid, and spermatid has to become a spermatozoa. The process is called spermiogenesis. Let us look at the process of spermiogenesis now. As we understand, the spermatid in the beginning is a circular cell, and then it is getting elongated, finally becoming a longitudinal type of cell with a long cilia, which is also called as the flagella, and the process of this conversion is called as spermiogenesis. If you look at the sperm, it will be having this long tail, which is considered as a long cilia, or you can call it as a flagella. It will be having the 9+2 structure of the microtubules, which will help it in movement, and this is all provided by the centrioles. And you can also mention that there is one tight sheet of mitochondria in the middle piece. Mitochondria, we know it is the storehouse of energy, that is the ATP, which is required for the survival of the sperm. We also see that in the head there is condensation of the DNA carrying the genetic component from the male, and this condensed DNA is so much condensed that it can be 6 times smaller than the normal somatic cell. Too much of condensation. And on the head we are having one acrosomal cap. This is derived from the Golgi bodies. We'll have some hydrolytic enzymes like the acrosin enzyme, and we need these enzymes to penetrate the zona pellucida for the purpose of fertilization. So what are we starting with? We are starting with the cell which is rounded and the spermatid in the lumen of seminiferous tubule undergoing spermiogenesis. You see, this Golgi apparatus is getting modified now and is forming the acrosomic cap, which we said will be having hydrolytic enzymes like acrosin helping us in penetration of zona pellucida for the process of fertilization. Then what about the nucleus? It carries the male genetic material and is undergoing condensation, so much so that it is densely condensed, almost 6 times smaller than the normal somatic cell. And what about the other component like mitochondria? It is giving us the ATP, the energy, and they are getting arranged in the middle piece, forming a tight spiral mitochondrial sheath. And then as we talk about the centrioles, the centriole towards the posterior pole is moving and arranging the tail in the 9+2 manner, a microtubular arrangement which will help to provide the sperm with a whip-like movement. But as we have mentioned, the sperms, they are immotile till they reach the level of epididymis. So before that, who was pushing them towards the epididymis? If they are immotile, it is the myoid cells in the seminiferous tubules which are pushing them towards the epididymis. And even after formation of the spermatozoa, there is one other process which is called as spermiation. So first thing is we have got the spermiogenesis, which is to be followed by spermiation. And what is spermiation? Spermiation is the loss of excess cytoplasmic structures which may be related with the developing spermatozoa, and that spermiation is carried out by the Sertoli cell, ultimately making the spermatozoa a very sleek cell because it has a long distance to travel for the process of fertilization. As we talk about the female reproductive system, we are going to focus upon the type of questions which they are asking and go through the details accordingly. For example, regarding the mammary gland, we have to understand it is a modified sweat gland and belongs to apocrine variety. So modified apocrine sweat gland. Then we have to discuss the epithelial lining. For example, the ovary. Ovary is lined by the simple cuboidal epithelium. And what about the uterine tube? As we talk about the uterine tube, we will find that it is having simple columnar epithelium. We'll be drawing a diagram ourselves to discuss the details, but some of the cells in this simple columnar epithelium will be having cilia also. So you can remember the uterine tube is lined by ciliated columnar epithelium. What about the uterus then? If we are talking about the uterus, it is having simple columnar epithelium. And when we talk about the vagina has the same epithelium as the esophagus. As we have mentioned, non-keratinized stratified squamous epithelium. We will also be discussing that vagina is a fibromuscular tube having some features which are just like the esophagus, but there are some differences as well. If we talk about the epithelium, they have the same kind of epithelium. Now, at the junction of the vagina and the uterus, that is the cervix region, there is a squamo-columnar junction which we have to mention. And let us look at a diagram which is a magnified view of this area. In this diagram, we are looking at the lining of the uterine cervix. The uterine cervix is lined by the same epithelium as the uterus we have mentioned. It should be simple columnar epithelium as is evident here. So this is the simple columnar epithelium which we will find in the uterus and also in the endocervix. So as we have marked it as simple columnar epithelium, what about the ectocervix? If we are looking at the ectocervix which is projecting into the vagina, it will have the same epithelium as the vagina. So what is the vaginal epithelium? We already know it is the non-keratinized stratified squamous epithelium as is evident here. Now, if we say this is the non-keratinized stratified squamous epithelium on the vagina as well as the ectocervix, it is understood there will be a junction which will be squamo-columnar junction, and that squamo-columnar junction you can see here. On the interior endocervix, we are having the columnar epithelium you can mention. And when we are coming to the ectocervix, then there is stratified squamous epithelium. That junction, as we are looking at this arrow mark, remains inside the cervix in a young female, but later it will be coming out towards the exterior, especially in childbearing age when there is eversion of the cervix. So that squamo-columnar junction which was lying on the inner side will be exposed to the outside in the childbearing age, and vagina has the acidic pH. That acidic pH is going to change the columnar lining of the cervix into the squamous epithelium. It is called as physiological squamous metaplasia, and it is quite normal. Means wherever we were having the columnar lining, it changes into squamous lining due to exposure to the acidic pH of the vagina. And later, when there is menopause, means the reproductive age is over, this junction, squamo-columnar junction, which came outside, goes back inside. So that means in the early female age and the later age, the squamo-columnar junction is on the inside, except for the reproductive age when it has come outside. Then we also should be careful. Whenever there is a metaplasia, there is always a chance of dysplasia, and dysplasia can result into cancer. So cervical cancer, especially in the females who are predisposed, we have to keep monitoring this area, the squamo-columnar junction, as they are prone to dysplasia and carcinomatous changes. Let us now draw this diagram ourselves and discuss it again. Now we draw this diagram ourselves showing the uterus here, and connected to the uterus, this is the uterine tube on one side, and we can talk about the uterine cervix here, endocervix, ectocervix, and then the vagina as such. When we are talking about the epithelium lining the uterine tube, it is understood that it is simple columnar epithelium and some of the cells will be having cilia. So ciliated columnar epithelium. But along with the ciliated columnar epithelium, there are some columnar cells which do not have cilia and they are called as the PEG cells. They are secretory in nature. So two type of columnar cells. In one type, there is ciliated columnar epithelium. The other is columnar epithelium but without cilia, called PEG cells and secretory in nature. Now, it is understood that the conceptus which is there in the uterine tube has to migrate towards the uterus because it is going to get implanted in the uterus. And to move this conceptus towards the uterus for implantation, there is some peristaltic activity of the uterine tube. Uterine tube has smooth muscles and those smooth muscles, they will be having peristalsis moving the conceptus towards the uterus. Plus, it is the cilia which are also helping to move the conceptus towards the uterus. Now, there have been some studies telling us about the importance of this cilia in the uterine tube. There is a condition which is called as Carter-Jenner syndrome, where we are having immotile cilia. A lady who was having Carter-Jenner syndrome or immotile cilia syndrome, some studies were done and these ladies, they have ectopic pregnancy. And it has been mentioned, since the cilia are immotile, they were unable to send the conceptus towards the uterus, and that is why the conceptus was implanting in the uterine tube itself, leading to ectopic pregnancy. So it means along with the peristalsis, the movement of cilia is important. And if it doesn't happen, that can compromise the fertility of the female. Now, do you have cilia in the uterus as well? No, uterus does not have the cilia. Uterus is having just the simple columnar epithelium. And that is the same you will find even in the endocervix. So, endocervix is also having simple columnar epithelium. What about the ectocervix? The ectocervix, which is projecting towards the vagina, will have the same epithelium as that of the vagina. So, we can mention here this region, ectocervix, is having the same epithelium as that of the vagina. And we know it is stratified squamous epithelium, non-keratinized variety. We also mentioned that there is one transformation zone which is between the ectocervix and the endocervix, and that transformation zone is squamo-columnar junction. So there is one transformation zone which is between the ectocervix and the endocervix, and it is a squamo-columnar junction. As we understand, the epithelium which is on the ectocervix is squamous, on the endocervix it is columnar, and this transformation zone normally in puberty is lying on the interior of the cervix. It's only after puberty, when there is a reproductive age, childbearing age, when there is eversion of the cervix during childbirth, that transformation zone is moving outside. The columnar epithelium on the endocervix is moving outside, but that columnar epithelium on the endocervix, as it moves outside, it is exposed to acidic pH of the vagina. Now, that acidic pH of the vagina is causing what is called as physiological squamous metaplasia, which is quite normal. And what we are having is the endocervix, a small portion which was lined by columnar epithelium. Since it came outside and it was exposed to acidic pH, it got changed into squamous metaplasia. And once the lady achieves menopause, this transformation zone again goes back inside. That means in puberty and in menopause, the transformation zone is on the inside. Only in reproductive age, it comes outside. Now, what is the problem? Problem is this metaplasia can change into dysplasia, can change into cancer, especially in the ladies who are predisposed to CA cervix. So we have to monitor such ladies for any such transformations. Now, we mentioned that the vagina is a fibromuscular tube and it has some similarities that of the the esophagus. Like esophagus is also lined by non-keratinized stratified squamous epithelium. But there are some differences. What are the differences? It comes as question. So you will mention the vagina has no glands. We understand that there are glands in the submucosa of the esophagus. Will keep secreting so that esophagus remain a wet mucosa. But if you do not have glands in the vagina, then how is it wet? There are some mechanisms, and one of that is the glands in the cervix. Their secretions keep trickling down into the vagina and keep the surface of the vagina wet and lubricated. So secretions keep coming from the cervix. As such, vagina has no glands of its own. Now we got one MCQ to deal with. Let us move on to that. Lining epithelium of vagina. And we already have mentioned, may it be the vagina or esophagus, it is non-keratinized stratified squamous epithelium. Now, as we find our answer, it is choice number C, and we have seen the same epithelium on the ectocervix as well. So, if this is the epithelium on ectocervix and vagina, your answer is choice number C. What about the endocervix? Endocervix, we have simple columnar epithelium. There are some glands also, secretory glands, keep giving secretions towards the vagina. And what about this choice number D? Where do you find this epithelium? If you say ciliated columnar epithelium, we have seen that in the uterine tube. And there we also had non-ciliated cells, which were called as the PEG cells. They were secretory cells. But as such, the answer for this particular question, we should keep it as choice number C.